So folks, between now and lunch we have a series of presentations, and I'd first like to introduce you to Associate Professor Luke Mosley from the University of Adelaide. Luke is a leading researcher in environmental chemistry and biogeochemistry. Luke's research focuses on the interaction between water, sediments and biota in aquatic ecosystems and how they are affected by natural and anthropogenic factors. He's conducted extensive research over the last 2 decades on the River Murray, lower Lakes and Coorong, where he has investigated the impacts of drought, salinity, acidification and nutrient cycling on these vulnerable systems. Welcome, Luke.
Thank you, and thanks everyone coming along today. And I'd just like to firstly say it's been a great project to be involved in. What I'm going to talk to you today about the nutrient dynamics project that I led as part of the HB program. So we started in 2020. So this program finished in, I guess, middle of last year. So it was prior to the flood. I will touch on some of the flood response right at the end of my presentation. But yeah, it was a really great experience. We had a lot of scientists working together across a range of disciplines, but also a lot of interaction with the community, which for me was a really valuable part of the program. And then we got to learn actually from the community, and many of the community members were passing on observations, which was really valuable and indeed helping us with the research as well actively. So many of you will be aware some of the background, I guess, for the HCHB program, with the Coorong South Lagoon in particular being acknowledged to be an undesirable state. And many of these problems can link back to nutrients, and I'll explain why that is the case. But many of the problems and poor ecological health does link back to primary factors of water quality and sediment quality. But I like to say, firstly, nutrients in and of themselves aren't a bad thing. In fact, we all need nutrients. Many of you would have taken nutrients in your breakfast today in the form of Weet-Bix or whatever you have for your breakfast. And it's really essential, for all plants and animals and a healthy ecosystem to have nutrients. Where the problems become is when nutrients become in excessive supply. And that's when, particularly in aquatic systems, with excess availability of nutrients, that really feeds algae and really drives a lot of those unhealthy cycles. So sort of like if you're getting plenty of nutrients, getting plenty of fertiliser, the algae can just grow and grow and grow, and that's when you start to have problems such as excessive algal growth, seen in this photo here, and as I'll describe, some very anoxic sediments. And that's known as a phenomenon of eutrophication, which is really just an increase in that organic matter, that algal supply to an ecosystem. So by adding nutrients, we can increase the organic matter and algal supply in an ecosystem. And internationally, when we look at the international literature on estuaries, really some of the key drivers of that are increased external loading of nutrients. As we develop our catchments, as we put more nutrient sources, whether that's farming, whether that's urban sources, fertilisers, those nutrients can eventually make their way to water bodies and increase that supply of nutrients and increase organic matter production. In estuaries, if we're not flushing the nutrients out to sea, they can accumulate. So we're increasing supply of those nutrients just by residing them in the estuaries for longer. So not flushing them out is a key driver, along with some other drivers such as climate change. Increasing temperature can increase production. So the project that I was fortunate to lead over that time period was the Nutrient Dynamics project. We had 4 main components. The first component was collating existing knowledge and data. There was a lot of disparate data and different data sources and different site names, and sort of the task was really to piece together all that historical, mainly water quality data, but also sediment data. The second task was really nutrient sources. Where are the nutrients coming from? Where are the stocks of nutrients in the system at any particular point in time and under different flow conditions? The third component is nutrient cycling and fluxes. So once the nutrients have entered into the Coorong, what are they doing? How are they changing? How are they moving between different parts of the environment? such as the sediment, water and algae. And the fourth was really taking that knowledge and trying to inform the broader assessment of nutrient removal options as part of the HCHB program, which I think will be talked about further today. And we were sort of informing on the nutrient biogeochemical side of those investigations. So today, as I think Alec mentioned, there's a lot of technical reports, a lot of technical information. So today I'm just going to share with you some of perhaps the key findings from those four components of the project. So we collated, I guess, made sense of all that previous data, collated it and plotted data. So that first task was really to look at nutrient trends over time. And we're fortunate, while there's been some gaps in the monitoring program here, for example, and here, there is a data set stretching back to the late 1990s, held mainly in state government databases. And this is just an example of total nitrogen in the water. And this is a site in the North Lagoon at Long Point, and this is a site in the South Lagoon at North Jacks Point. So you can see the value of having these long-term data sets in this example, where the millennium drought here comes along, and we start to see, particularly in the South Lagoon, really some extremely high total nutrient concentrations. The Australian Water Quality Guidelines down here in yellow, so we're getting over 10 times what the Australian Water Quality Guideline for total nutrients are. And I'll explain some of the reasons for that, but essentially it relates to lack of flushing of those nutrients. The nutrients are just accumulating in the system. And there's a technical report and also a scientific paper if you want to look more closely. When we started to plot, I guess, the nutrients versus salinity, we were interested in how nutrient concentrations relate to salinity in the Coorong. And it's fair to say that, firstly, total nutrient concentrations in the freshwater sources are much lower than the Coorong Lagoon concentrations for the most part. So you can see here, this is a plot of salinity. So the lower lakes inflows are down here, below about a salinity of 1. And you can see once you get over seawater salinity here with a seawater of 35, so now progressing down the Coorong, and this is all the data combined, you can see as you get to, say, double salinity, the nutrients really increasing and so on, and up into those extreme salinities in the millennium drought, you've got those really high total nutrient concentrations. So there's very strong relationships of total nutrients and chlorophyll with salinity. As I mentioned, the Coorong is, as Nadine mentioned, a reverse estuary. The evaporation of water, so increasing the salinity, can also concentrate nutrients. So while the water evaporates and leaves salt behind, it also leaves total nutrients in the water as well. So they can accumulate, and if you're not flushing those nutrients out to sea, they will keep accumulating. And some of the work we did, and probably no surprise to this audience, that the River Murray influence is extremely important in influencing the North Lagoon in particular, but to flush the South Lagoon, you need some very, very large River Murray flows, and I'll show you some data at the end of my presentation on these recent floods that occurred after this program ended. And the other final point is that dissolved nutrients are generally at low concentrations, so any available dissolved nutrients are what the algae take up. So any of those dissolved nutrients that become available in the system, they're rapidly taken up by algae, and they're driving those high concentrations of algae. So that was just a little bit of a snapshot of the water quality. Water quality and sediment are closely linked in shallow systems, and as we've shown with the nitrogen increasing with salinity, the nutrients in the sediments of the Coorong also increase with salinity. This shows you total nitrogen here, and the red and yellow colours are high concentrations of nutrients grading to sort of more bluey, greeny colours, which are lower concentrations of nutrients. So there is a gradient, and these deep basins of the South Lagoon are really good accumulators of nutrients. We find very high concentrations of nutrients in those areas of the deeper basins. And some of the calculations we did, there's 50 50 times more nutrients in the top 5 centimetres than in the water column. And really the reason for that, I've talked about the algal production, the algae taking up nutrients, but when that algae dies, that algae settles to the sediment. So that those nutrients then come down to the sediment and build up the stocks of the sediment. So as we've talked about with algae not getting flushed out of the system, that means that the sediment also accumulates nutrients. And one of the, I guess, things that can happen when you get a lot of organic matter, a lot of dead algae going to sediments is you start to, when the microbes or the bugs in the sediment start to break down, they consume oxygen and you can get very anoxic, black, oozy sediments. So many of you that have walked around the Coorong, this is on the shoreline here, you'll encounter this mysterious, what we call anoxic black, monosulfidic black ooze. And you know, there probably was a bit of it around, I think, in the past, but now we're seeing really dominance of this sediment type, which is really an unhealthy, it's very anoxic and a very unhealthy sediment type in much of the system. If the nutrients just resided in the sediment, you know, it's still not great, but what is actually happening is those nutrients are then being recycled back to the water column. So as those microbes, as I've described, break down that organic matter, sort of like compost in your garden, you add organic matter, that microbes in your garden break it down and that's being released for your plants. And it's similar here, but the nutrients are now breaking down in the sediment and then they are being fluxed back or diffusing and being transported back to the water column. So it's sort of a cycle and it sets up what we call a vicious cycle where you get a lot of algal production, that dies, settles to the sediment, breaks down and goes back and feeds more algal production. So the cycle can just keep continuing if it's not getting flushed out of the system. And we find very high dissolved nutrient concentrations in support of that in the sediment. And we find unhealthy processes going on. One of the key processes that we like to see is denitrification, and that's a way that nitrogen can actually go out to the atmosphere as a gas. And the South Lagoon is probably one of the few systems we've seen where you don't really get any significant denitrification. It's a key component that's very limited in much of the system. So that again means that nutrients can accumulate in the system if they're not getting lost out of the system. We do see some positive signs where there's aquatic plants, and Michelle might talk more about that, but where we've got aquatic plants, you can get nutrient uptake and you can get these lighter coloured conditions in the sediment, so starting to break down that black ooze. So the aquatic plants definitely have a very positive influence on the nutrient cycles. And so do benthic macroinvertebrates. So this was a component done by Professor Sabine Dittman and her PhD student Orlando as part of our project. And one of the features, I guess, of these anoxic black oozes in the South Lagoon is we don't see invertebrate burrows. So this is some in the North Lagoon. You can see these tubes extending down, these macroinvertebrate burrows, but you can see this black oozy stuff in the South Lagoon. It's really lacking macroinvertebrate burrows. And when we look at the abundance, and Sabine, I'm sure will talk more about this, this is a site near the Murray Mouth, and this is at Salt Creek, and this is abundance of these macroinvertebrates of different groups. And you can see that once the salinity gets over about 60, on the right-hand axis here, we're losing all the organisms that actually burrow in the sediment, and there's just really surface deposit feeders left, chironomids and things like that. So why is that significant? Well, in a healthy ecosystem, all these organisms that biota, worms that dig, they function in several ways. They bring oxygen into the sediment, those burrows. The oxygen can travel down in the water deep. They process organic matter, so they can break down organic matter and can start breaking down those high concentrations. But what we see, particularly in the South Lagoon as we progress along it, is we start to get less of the healthy, what we'd like to see, nitrate, and we get more ammonium, and we get more organic matter building up in the system. So as we lose macroinvertebrates in the system, our sediment nutrients are in an unhealthier state. So one kind of quite cool experiment that Orlando did as part of his PhD was he took some of these really unhealthy sediments. So prior to this big flood, we couldn't have actually put macroinvertebrates into the South Lagoon because they would just die because the salinity was too high, as I said, over 60. So at this time, what we did was we collected these sediment cores from the South Lagoon sites that had high salinity, high nutrients, high sulphide, and we actually transplanted them in the North Lagoon where the water salinity was low enough that these burrowing organisms could survive. And what we found over a period of only 4 weeks was as organisms started to burrow into the sediment, they started to break down that organic matter and start transforming those nutrients into healthy state. So really in a short space of time, by introducing macroinvertebrates, we were able to transform that unhealthy sediment. And I'm sure Sabine, I won't steal Sabine's thunder, but the large floods have showed now what this experiment gave a precursor to, essentially. Because the salinity is now below 60, there has been some of this activity observed. But we'll leave you hanging, wait for Sabine's presentation. I guess as part of this, and I'd just like to say this is really just from our nutrients team, so it's only really one part of the decision making. Is that 5 minutes? 5? Yep. Yep. Just checking it wasn't the one.
5 minutes to go.
Yep, that's plenty of time. As part of this was, I guess, from our scientific perspective, but there's a whole range of other decision making that will go on, but into what nutrient removal options might be moving forward. But as I mentioned at the start, flushing for the Coorong is particularly important, just the nature of its system and its vulnerability and the changes in flushing that it's had over time. That by able to flush the system, you can remove a lot of that algae, so stop it accumulating in the system and get it moving out to sea. How you do that is open to debate. And we've talked about increased River Murray flows, and there'll be other options being scoped, such as infrastructure out of the South Lagoon. Secondly, if you reduce the nutrient loads entering the Coorong, that is highly beneficial. But we've got a 1 million square kilometre catchment with a whole lot of politics, and trying to do nutrient removal programs at that scale, I don't think it's been done successfully, certainly anywhere in the world at that scale. It's very difficult to do large-scale nutrient removal options in a catchment as big as that, with all the political boundaries and things. We looked at artificial oxidation of water and sediment. Like I've talked about how that black ooze is very anoxic. There are ways you can oxygenate it, but you're talking, oxygen tanks, aerators, all sorts of complex, expensive infrastructure, and to do it in a 100-kilometre long lagoon at scale, and it doesn't address the primary drivers of these problems, wasn't deemed a great option. Macrovites and benthic macroinvertebrate restoration, that'll be, I'm sure talked more about today, but these things are highly beneficial, but we've got to have the underlying conditions right where these can really thrive and really start to really take over and dominate the processing of nutrients and get it away from these algal sort of mats and algal phases. Internationally, people do harvest algae, but again, it's sort of like the ambulance at the bottom of the cliff. You're going to have to keep doing this for perpetuity if you're not fixing the problem. You're just picking it up when it's become a problem. And, certainly at a small scale, hostile, trying to cap the sediment and remove it, but does have its own impact, that sort of activity, and probably only applicable again at small scales where you've got some particularly bad sediment and maybe some areas that are priorities. So really what we found was, that improved lagooning, flushing and connectivity is really important. And just to show you this graph again, what we're sort of, talking about is really not existing out in this space. It's really trying to get the system down here where the nutrients are a lot lower, but macroinvertebrates and rupia can do well down in this range, and then trying to enable them to further improve the system. And not going to have a lot of time to talk about the floods, but obviously I was really excited when the floods from a scientific perspective. I'm sorry if anyone was personally affected by the flooding, but it was a really great opportunity for, I guess, scientists to study the effects of the flood on the Coorong. And what we've found is, while there's been some impacts near the Murray mouth, as Adrian said, where there's a high amount of fresh water that's got high nutrients coming off all the land in the Murray Basin, the South Lagoon has actually had a massive improvement. It's had a 30 to 40% reduction in total nitrogen and total phosphorus. These large floods historically performed a really key role, and we've actually got data from the 1970s flood for the Coorong that showed similar effects, a really big flushing effect, but you need these large floods. Trickling water over the barrages helps a bit in the North Lagoon, but these big floods can really push into the South Lagoon. But obviously, as has been touched on today, the median, I think it was Nadine's presentation, no, was it Adrian's presentation? The median floods here. The flood that we got was above what the median was, but the median was way higher than we're normally getting. So we used to get those floods, very regularly, and we're not getting them. So there is still rationale to pursue other actions because we can't rely at this stage on, those big flood events. And I'd just finally like to thank, it was a huge team effort. I've probably missed some people off here, but some of them are in the audience, and thank you.